And it's now over to you, Erika.
Oh, thank you very much. So hello, everyone. My name is Erika, and I am a part of the DMPK team. And today we're going to be talking about the use of animals in drug discovery. I just would like to make it as interactive as possible. So if you guys have any questions at all at any moment, just stop, just put your audio on and just ask away. I'm on my laptop, so I may not be able to see the questions coming through, but Laura, you're going to be helping me through it, okay? So the title of this presentation today is The Use of Animals in Drug Discovery. And I am an in vivo pharmacologist in the DMPK team. And very recently, I became an in vivo DMPK trainer, okay? We have some learning objectives to go through the presentation. So why are animals needing for research? Who are the scientists who work with animals? And animals have rights, and there are strict regulations in place to ensure ethical usage of animals. Of course, that when you're going to be talking about legislation, I will be focused more in UK legislation, but it is also very similar to European legislation. So I know that for different countries it's going to change, but I'm going to give the point of view of how we work with the animals here in the UK. And just a definition before we really start the presentation. So as I mentioned, I'm part of the DMPK team. So what is DMPK? So DMPK stands for Drug Metabolism and Pharmacokinetics. It pretty much says you're going to study how the body breaks down molecules. When we talk about DMPK, we have two main topics that you're going to be focusing. First one is pharmacokinetics. So we normally call it PK. What I want you guys to remember about PK is what the body does to the drug. So what do I mean? What the body does to the drug? When you take a compound, a pill, you're going to go through a lot of process. So absorption, you're going to get it through your mouth, you're going to go to your stomach, and then you're going to distribute through your body. You're going to reach your liver, where most of the metabolism is going to reach, and you're going to be excreted from your body. So when you talk about PK, we have this admin, as you can see here in the slides. Let me just get my pointer, if I can see it in here. Yeah, here, admin. And sometimes you can see a T at the end here, that it also includes toxicological studies that we don't do here in the DDU. But if you guys sometimes see, instead of admin, admet is also toxicological studies. But what I want you guys to remember from PK is what the body does to the drug. And for pharmacodynamics, PD, it's pretty much what the drug does to the body. When you talk about PD, it's going to normally be a sick animal. So you're going to be talking about an infected animal, an animal that presents some kind of disease. And PK is a naive animal. What I mean for naive animal is an animal that is clean. There is no disease. It's a clean animal just to see what the body is going to be doing with the drug. And of course, they interrelated. So the PK, it starts with the drug. So you're going to take the drug, which you're going to reach a blood concentration, and you're going to drive a concentration in the receptor side. And the connection is for the PD is the receptor side. You're going to drive a pharmacological response that you're going to result in a clinical outcome. So the link between pharmacokinetics, PK, and pharmacodynamics, PD, is the receptor site concentration. That pretty much means, can the drug do what it has set out to do? And I know that you guys have been using Mentimeter in the last talks, and I really hope I can switch for the Mentimeter page here now. And I would like you guys just to take part in what would be your thoughts on animal research. I just would like a word or a few words, what you guys think when you think about animal research. Okay, let me try to move to the monitor here now. Can you guys see my screen?
No, it's still your presentation.
Let's see. Shall I stop sharing the presentation and share the Mentimeter then? And let's see. Yeah, Lauren, you think that is all right?
Yeah, yeah, do that. That'd be good.
Let's see. Can you guys now see my Mentimeter screen?
All good.
Good. So I just would like you guys to write something. You can write as many words as you want, or phrases or anything. What are your thoughts on animal research? I know you guys are used to the Mentimeter, so if you just join the ment.com and put this code, you're going to be able to take part in this. There you go. Something starts to come up already. Necessary, yes. Ethically problematic, it's a really good one. It's very difficult to be talking about animal research, especially here in the UK, and you're gonna go through it. Very important, yes, it is very important to test our drugs in the animals. Crucial, mammals, yeah. Inescapable, I like that one. Yes, it is inescapable. Morally wrong. Well, let's see what we can talk about it. Clinical trial, we're going to talk a little bit about it because here for animal tests, we are in the preclinical area. Clinical trials is more when we go to humans trial, but we're going to talk a little bit about it. Can be reduced, a very important one. We always try to reduce the number of animals we use here, and that is very important. It is very costly as well. Very nice. A lot of words coming through. That is real. Rat models, yes. Disease model. Necessary evil, yes, it is. Indeed. Yeah, that is dose optimization. Test on criminals instead. Yeah, we're gonna be talking about it a little bit. But yeah, I really like what you guys have put through. Need better alternatives. Yes, animal cruelty. I was expecting this one to come through as well. I really like, if you guys at any time would like just to unmute yourself as well and try to make it more interactive, we can try to do it, okay? So thank you very much for everyone that answered this. Some more is coming through. Similar to humans, yes. It's a safe barrier, yeah. Cool. I think we have reached a good number here. And I would just ask you guys to still with me in the Mentimeter, because after this I have another questions for you. And I hope I can move for my next Mentimeter slide. Can you guys see this next one?
Yep, all good.
Okay, so my next question here for you guys is, why do scientists use animals in research? You can answer more than one as well. Yeah, that's really good. So yes, we do use animals in research for different ways to try to answer different questions that we have. But yeah, some of them is to understand the body in health and illness, to safety testing medicines that can be different kind of medicines, to do research and development of medicines. And yes, I'm very happy that anyone have replied, because they hate animals, because sometimes people associate that the people that work with animals, they hate animals and they do it because they enjoy. And I can tell you that is the other way around, really, because we really like the animals, but it is very necessary to do. And we don't like to do it, but we have to. And we try to minimize the suffer. We try to minimize everything we can to make this as smoothly as possible, if I can say in that way. But yeah, we really care about the animals and we don't hate them. We just do this because we have to. Okay, so I think I'm going to stop to share again and try to go to my presentation again. Sorry for that, but as you can see, I'm not very good with this IT kind of stuff. And I hope you guys can see my presentation again.
Yep, can see your talk.
Okay, so why are animals used in scientific research? So we can see here in this picture that we have a human here and an animal. And you can see that we are very similar. Not even looking inside first, but just if you look outside. We both have a pair of eyes, we have ears, we have a kind of a nose, you know, a mouth, we have arms, we have legs, and we are very similar from the outside. But also, if you look inside, we look very alike. In fact, might share more than 98% of the DNA with us. So as you can see here, apart from the DNA that we share that is very similar, we have a pair of lungs, we have our liver, we have our gut, we have the heart, and as you can see, exactly the same in the animals. So we are biologically very similar to the animals. And why we use animals? So the animals, they are susceptible to many of the same health problems than us. So we can induce in the animals different kind of diseases. For instance, we can induce a cancer, we can induce diabetes, we can make the animal infected with some parasite. That's more our area here at the DDU. And they are going to present the same kind of symptoms that we will present as well. So it makes a very good model to study the disease progression as well as how the drug is working in the body. Something else that is very important to say is the animals, they are very easy to care. What do I mean by easy to care? So they are very small in comparison to a human. So we can put a lot of animals inside of a room. And we can care about them with the diet, so we can control what they eat, what they drink. We can control the temperature of the room. We can control the light. So the animals we have here, they have 12 hours light, 12 hours in the dark. So we can control it very well. And in the Mentimeter, someone said, test your drugs in the criminals instead. Let's imagine if it was legal, if it was ethical to do so, would be very difficult to do that and try to control the diet, try to control the temperature, fit a lot of people inside of a room with different body weights and this kind of stuff. So even if it was legal, which is not the case, would be very difficult to try to test drugs in humans. Okay, something else that is very worth mentioning here is the animals, they are very easy to reproduce as well. So the mice, they reproduce very fast and they have a lot of baby mice that is very helpful for us when we need a large amount of animals to be using in our research. Okay, so animals, they are specially bred. What do I mean by that? So you can say to me, oh, I live here in this place and I just saw a family of rats that they just gave birth. I'm gonna get some of these rats and I'm gonna try my medicine that I'm doing in the lab. Can I do that? And I will say to you, please don't do that. It is extremely unethical and you have no control of your animals. So the animals we have here, they are specially bred for this purpose. So they have control of light, they have control diet, they have everything they need in this environment. If you get a rat or a mouse that you see on the street and try your drug, you're gonna be pretty much just killing an animal, and you're not gonna have a good result from that. So please don't do that. Yeah.
Can I just ask, there's a question in the chat, so it
Yeah,
refers to what you've just been speaking about.
Yeah.
We've got a question saying, do they develop diseases more quickly, usually, for example, age-related diseases?
Yes, they do develop more quickly, and I can give you an example here. So we work with a disease here in particular at the DDU. The name is Chagas disease. It's caused by a parasite, Trypanosoma cruzi, that is very typical from South America mainly. This disease, what we want to treat is the chronic phase of the disease. And for humans, you go well, you're infected with the parasite, and you don't know that you have the disease until 35 years later, when you became it to treat you, because we don't have a drug that works in the chronic phase. We use here the animal model, and instead to wait for 35 years for an animal, because the life cycle of the animal is a lot shorter as well. So here, after 100 days, 93 days more or less, our animal is chronic. So yes, it's a lot faster. Because if you think about age average of an animal, depends of the strain, but would be around 2 years more or less, which is also very important for us because we can follow the animal through their life if you need a drug for chronic phase or this kind of stuff.
Erika, can I just ask this a couple more questions?
Yeah, just fire them through.
Yeah. So the next question is, would it ever be possible to envision a clinical trials future where animals are not used?
So when you talk about clinical trial, it's more when you talk about humans. Here we are in the preclinical phase, just to clarify this first. Yes, it may be possible in the future, but I cannot really see this far ahead. Actually, in fact, FDA just have put some months ago saying that they would not allow animal testing anymore. But I don't think this is going to be happening soon-ish, because we need to prove it's a legal requirement. You're going to be seeing the next slides to test our drug in at least 2 different kind of animals before go to clinical trials that goes to humans.
Cool. Thank you very much for that.
Is there any other one?
There's more in the chat. So how much difference does that 2% difference in rat DNA make when it comes to testing drugs?
The DNA in particularly here, because we don't do DNA kind of genetic drug discovery at the DDU. So the DNA wouldn't make a lot of difference, but we have other factors that makes a difference. And one that I can mention here is, for instance, the liver blood flow, that in mice is around 6 times faster than in humans. So when you give a drug for an animal, a mice, for instance, it's gonna be cleared by the liver 6 times faster. So we have to take it into consideration. And there are ways that you can block this fast metabolism in the liver if we need to, but it's just some things that we need to keep in mind to consider when we get our result back.
Cool, thank you very much. So that's all we've got in the chat, so you may continue, so thank you for that.
Yeah, thank you very much for the questions, guys. Really enjoyable to be like just getting the questions as soon as we go through, so we can clarify everything and everything goes more smoothly from there, okay? So, okay, we agreed in here that we're not gonna test our drug in the mice that we just saw on the street. And other thing that I want to mention here, that it's illegal to use animals if there is any other way of doing research. So what do they mean by it? Before we go to the animals, we need to have some kind of proofs that you're gonna be worth to put it into the animals, because we always have to think that it's alive. When we put it in the animals, eventually we're going to terminate the animals. That is what we have to deal with. So we have to prove that it needs to be there. And how can we prove that? We have different of non-animal methods. One of them is in silico that you guys have heard about. So lots of mathematical models to make sure that this compound is safe enough, it is maybe soluble enough that you're not going to be metabolized too fast and disappear from the body. So when you take your blood from your animal, we're not going to have any compound in there. And after in silico, that would be more the mathematical methods, you go to in vitro. In vitro is when you have your cell in a test tube, pretty much, or a piece of your liver in a test tube, and you test. You do a lot of tests before it goes to the animal in vitro. Something that is important to mention here as well is when we do in vitro tests, and if you're using a parasite, for instance, or if you are using a piece of liver or some blood, it came from an animal eventually passing the line. So even with the in vitro tests, some of them, it came from an animal, but it is diluted. We're going to be using that for a very long time. And we can use one liver to do a lot of compounds, for instance. But even the in vitro, we use an animal eventually down the line for that, okay? So here in the UK, our animal research is regulated and inspected by the Home Office. I'm not going to be talking a lot about legislation because it would be very boring here, but I would like to mention a lot of things that is very different than the country I came from. I'm from Brazil, and I've been working with the animals in Brazil, and the legislation that we used to have in there is a lot different than what we have in the UK. So first of all, license. Here in the UK, to be working with an animal, if you're gonna touch a live animal, you need to have a personal license. So what does it mean? When you start to work here, you go through a course and you're gonna learn a lot about the animals. You're not gonna practice in any of the animals because it is illegal. You cannot practice any of our procedures with the animals. We can practice when they are dead, but if they are alive, you can. It's against the law. But you're gonna go through the course, you're gonna learn a lot about the animal symptoms that they may have, what you're gonna do if something unexpected come up, and how you're gonna deal with that. Apart from the personal license that every single person that works with the animal in the country they have to have, you have to have a project license as well. So remember, in the beginning of the presentation, I said to keep in mind about the PK and PD. So PK is the naive animals. PD is the animals that are infected. So if you are working with both of these animals, you're going to have two different project licenses, and we can just do what your project license states. So you can just, for instance, administer a drug in your animal orally if your project license says that you can. You can just take certain amount of blood from your animal if your license says so, you know. And you have a certain number of animals that you can use per project. Sometimes it can be amended, but they always try to make use as little as possible and try to minimize everything. And when you have to use an animal, get all the information you can from that animal. Apart from personal license and project license, the institution that we work, they also have a license. So here, the University of Dundee, they also have an establishment license where they can say that you're going to provide rooms, that you're going to provide the staff to look after the animals, that you're going to have an autoclave to autoclave the food, the water, and have a space to you to work. For instance, if someone see you doing a procedure that is not listed in your project license, they can take out your license and you cannot work with the animals anymore. Or even if you are doing something very wrong, they can even close the establishment license and say, listen, this person is doing something that is against the law, and we're going to close the site and we cannot do any animal work here anymore. And, drug discovery process, you need the animals eventually. So would impact a lot in all of the projects. We have some regulated procedures. So I mentioned before in the project license, you can just do what it is written in the license that we can, and you have to follow it. And we have some code of practice. So for instance, we work with infected animals. So if something happens, if you stab yourself, if you have some splash in your face, what you're gonna do? If an animal biting, or what you're gonna do? What is the procedure that you have to do if something that is, out of your control just happen? And you have always to be up to date with all of these procedures. Inside of our animal unit, we have a lot of different people that work there for us, but not actually for us. They work for the animals to see if everything is doing all right. I will just mention a few of them, but for instance, we have a vet that you're going to be there and you're going to advise you if your animal is present some symptom that is something you are not expecting from the disease. She's going to advise you and going to look after, and maybe going to give a treatment for that animal to try to minimize the suffer that the animal may be having. This kind of stuff. And we're going to have to evaluate if the treatment the vet is giving, you're not going to interfere with the treatment you're going to be giving by the animal, and this kind of stuff. NTCO is a very important one as well, because this is a competence officer, is a trainer pretty much. And they come to evaluate us to see if the procedure we are doing is correct, is up to date, is in the best way it can be done. And it is done with us for every single procedure we do once a year. So we're gonna bleed an animal. So these officers are with us to see if you know how to do that, and if you know that in a nice way, and if you take the amount of microliters that we have to, that is written in our license, and this kind of stuff. The home office sometimes can just appear in the establishment you're working and just ask to, not even ask, just appears there and see what you're doing and start to question you why you're doing this way. And you cannot say to them, listen, I don't want you to see, because it is all regulated by the home office. So you have to show them and explain what you are doing, why you are doing. If you are not doing something, why you are not doing that. And if they think you're not good enough, they can also revoke your license. They can say, listen, you're not good enough to be working with the animals. You don't care about them enough. Or this procedure, there's different ways to do, and they can just take your license. So the home office here in the UK is very strict, and we have to work by the laws.
Erika?
Okay, yeah.
So you've got a few questions in the chat.
Yeah.
Mikkel, though, your question, that will come up in the talk, so I'm not going to ask Erika that one just now.
Okay.
But the next question coming on is, can molecular dynamics simulations cover up for animal testing?
They would not cover up. They're going to be added on it. So of course, you're going to do it first and see which kind of interactions we're going to have, and if it is good enough to proceed down the line to go to the animals.
Cool. And we've got a question here. I found it actually quite an interesting one. So we've got, I understand that there are generalized species for testing, like rat, mouse, or dog, but are there noticeable differences between subspecies of those animal types? So we've got, for instance, would a drug tested on a pug also be equally effective on a sausage dog, given they are roughly the same size?
Well, we have even more specific differences about the sex. So sometimes what you have in the male dog is different that you're gonna have in the female dog. So yes, there are a lot of difference, but you have to see the correlation between them. So I will be talking about it through the presentation, but just to clarify right now, If your model of disease, for instance, remember here we are in the beginning of the drug discovery, yeah? So it's this preclinical phase. But if you're going to be testing our model of disease, for instance, in a female mice, our naive animal, you're going to also be female. You will also be the same species. For mice, for instance, you're going to be using bulb-C for the naive animal, bulb-C for the treatment animal. But yes, there are a lot of differences, even between sex. So yes, between strains or even different kind of animals. Yes, a lot.
Cool. And we've also got another question. Is the stress that an animal may suffer also evaluated?
So no, not here with us. We don't evaluate stress. We could do that. We can measure cortisol, for instance. We don't even do it. We don't actually do it in here. Some institutions may do. What we always try to do is minimize the stress. I don't know if you guys are aware, but in the past, everyone that worked with the animal, they would pick up the animals by the tail because it's just easier. You know, you're not gonna have a bite from the animal, but we cannot do that anymore because it has been proved that it's very stressful by the animal be picked up by the tail. So what we do, we cup up the animal. So we have like toys inside of the cages of the animals. And then one of the toys is a kind of a tunnel and the animal enter and then you just close with your hand and just manipulate from there. So we try to minimize the stress, but we don't measure the stress here at the video.
Cool, excellent. And we've got an out of there question that read somewhere that the most expensive domestic animal in the world is a lab mouse. Do you agree with that? And if you do, what do you reckon is the total cost of a lab mouse?
Well, I don't know if it would be the most expensive in the world, but yeah, it costs quite a bit because it's not just the cost of the animals. We breed some animals in-house, but we mostly buy our animals from other institutions. For instance, Charles River that we have in lots of places in the world, or different labs. Jack Labs, for instance, is another one that everyone knows about. I can tell you bulb-C mice or a C57, that is a strain that we use the most, they cost around 20 pounds per animal. And you may find that this is expensive already if you think about one animal. But when you include in this price all of these people that are employed to look after the animals, the food that the animal is going to be eating, the water, and all the care would be a lot more than that as well. So I don't know if it's the most expensive in the world, but yeah, it would be very expensive. And here we are talking about mice because at DDU, as I said, preclinical, we work just with mice and rats. We are not even talking about the dogs or monkeys that go, or pigs, that are bigger animals. You're gonna eat more, and you're gonna be looking after a lot more in big spaces than ours here. Yeah, is that it?
We have one more question, but do say if this is going to be covered further on. So we've got, how do you design a study with animals taking into account differences that have just been mentioned? For example, differences between subspecies, gender. Can you somewhat accurately quantify these differences up front and then evaluate their effect on the drugs PK and PD, and finally extrapolate that to the humans?
So extrapolation for the humans is a difficult topic, but what we tend to do, we're gonna go with a sex, for instance, our PK and our PD. So let's imagine here we're talking about bulb-C female, that's gonna be doing. When you're gonna run an MTD, that would be a maximum tolerated dose, we're gonna be running with, for instance, male rats and see how they're gonna behave. And we have some softwares for extrapolations that we can use and can give us some kind of idea, but shouldn't be massive differences than what we would be expecting and from what we know from literature, from different insects and species. But the thing is, we need to test at least in 2 different rodents, for instance. So differences we're gonna have, but we are aware of these differences, and we're gonna be making our calculations that would all be included.
Excellent, thank you for that, Erika.
Yeah, that's fine.
Continue on.
Okay, cool. So why are animals used in scientific research? So we're gonna test drugs for either veterinary or human, right? So we're gonna be testing the drugs that are gonna be used for veterinary uses in the animal, and this would be our clinical trial already because it's the same species that we want the drug to be. Or if you're gonna be a drug that's gonna be for human, we're gonna have to test in the animal model first and then go to humans. But must be tested in animals before go to humans if this medicine you are looking for is for human. And it is actually a legal requirement for medicines that they are testing 2 different species of mammals before clinical trial. And I have just mentioned the questions this before. I have just put here a pipeline that we use quite a lot, and I know that you guys have seen this with Lauren before, just to say that you guys know that Discover Science is not really in our kind of pipeline, but just to say that the animal, we are in this from hit to lead to preclinical candidate selection. This is where in the line you're gonna be using animals for. More animals are gonna be used here in preclinical development, and here clinical trials that you're gonna be talking about humans test, not animals anymore, okay? So what do we need to have before going into animals? So we have our compound. Our friend chemist just came here and said, look, Erika, I have this beautiful compound. Can you put this in the animal? Because I'd like to see how it goes. And I would say, hell no, cannot go to the animal before we have some kind of tests before. So which kind of tests that we need to have before go to the animal? So first one, our compound, they have to be potent and selective. So what do I mean by that? Let's use the example here of leishmaniasis because I believe Lauren been talking about leishmaniasis with you guys. So leishmaniasis is a parasite that lives inside of the macrophage. So our compound needs to be killing the parasite inside of the macrophage, but not killing the macrophage. So it means that our compound is potent and selective. So I gave this compound that the chemist just gave me, and I passed to the biology team, and they test and they say, yes, this compound is potent and selective. So it kills our parasites and don't kill our cell. So good. You're not going to be toxic for us, but you're going to be toxic for the parasite. That's what we want. After that, we're going to do some in vitro DMPK. So we're going to need to see if our compound is soluble. Going to be dissolved, because if our compound is not soluble and you're going to give this to the animal, you're not going to dissolve in the stomach. So what's going to happen? You're going to take the blood of the animal and not going to see anything because the compound didn't even dissolve in the stomach. Is our compound permeable? You're going to go through the cells. We need to know that before give to the animal. And is our compound stable? So the liver didn't break this compound and make it disappear. So these are the 4 main experiments that we need to have before put our compound into the animal. So if any of you guys would be seeing something that wants to work with the animals, this is the kind of experiment that must be done before we put something into the animal. If it is not done, I would say that is very unethical to be doing any animal study, okay? If you have these experiments and they look good, because if the results here are not good, we're not going to put into the animals as well. They have to be good to give us some directions what's going to be happening in the animals. And if they are good, then yes, we're going to do an in vivo PK with the animals, okay? Something that we work with is the NC3Rs. So the NC3Rs are the National Center for the Replacement, Refinement, and Reduction of Animals in Research. So we need to follow the NC3R rules to work with the animals. So I'll give you just some examples of replacement. As I mentioned, we need to do mathematical tests, in silico tests before it goes to the animals. We need to do in vitro tests, as here they say tissues or cells, before it go to the animals. So this is our replacement. For refinement, we need to improve the welfare of the animal. So we put some toys inside of the cage so the animals, they are not stressed. We don't overpopulate the cage with a lot of animals that they don't have spaces to run around or to hide, because this is their behavior. That's what they like to do. So we have a number of animals that can be put for a cage. For instance, a cage of 40 by 40 centimeters, we put 6 animals. A cage by 20 centimeters, 3 animals. So remember that these animals, they are quite small, 20 grams more or less. So this is some kind of examples for refinement. And reduction, we try to minimize the number of animals that we use per experiment. And I will be giving good examples for reduction through the presentation. So how does it really work? So remember, we have our compound that's potent and selective, that pass all the in vitro DMPK, and now we're going to put our animal for the first time into the animals. We're going to have to select which animals we're going to use. So here, just put some images of different animals we use here, and these animals are the ones that is most studied and used for drug discovery. So this one is a BC. Is a small, very docile kind of animal. Lots of research are done with this kind of animal. We have this one here that is the C57 black, and this is a wild type kind of animal, so it's a little bit more aggressive. They're gonna try to bite you whenever they have the chance. This is a hamster, looks like a bear even. It's very, very cute. But there's a problem, they don't have a tail. So when you take the blood from an animal, if this one doesn't have a tail, it's a kind of a problem. We can take the blood by the saphenous vein. That is a vein that the animals have in the leg, but require at least 2 people, one to be holding the animal and the other one to be doing the cut and take the blood. And if you're talking about an infected animal, you need to be very careful with that as well. And this one is a rat that looks very similar to the bulb C, but is a lot bigger. Can go to 600 grams easily, even more. So, and they are very docile as well. So for our PK experiments, we use 3 animals per experiment. So it's not very statistically relevant 3 animals, as you guys may know, but if you have some problem, we can see, and we try to reduce the number of animals. So what we don't want to be doing here is to be using 1 animal, and then you cannot trust your result because you don't have anything to compare with. And if you have an outlier, for instance, how are we gonna know? So with 3 animals, has been proved that you can see all the curves you're gonna get from that animal. And if you have an outlier, for instance, you can pick that up. And even if you need to exclude your outlier, you can, because then you have another 2 animals to be comparing with. If you are using 2 animals and you have 1 outlier, you're gonna not know which one is your outlier really. So 3 is what we stands for and works well for us. Because if you use 2 or even 1, and then you're gonna have to repeat that, that means you're gonna be using more animals down the line. So for PK, naive animals, we use 3 animals per experiment. We are able to take small amounts of blood from these animals because we have a very good mass spec. So with 10 microliters of blood, we can see if you have some drug in there. So what we do normally, we inject our compound in the animal and we take some spot bleeds. That is just make a hole in the tail, take 10 microliters, and we can take up to 10 time points from 1 animal, which is massive. It's really, really good. And that is just possible because we have a really good mass spec. is in the legislation that we cannot use more than 10% of the blood volume per animal. So we always have to keep it in mind to see how many time points we're gonna take and depend of the body weight of the animal. And as I've been mentioned during the presentation, same sex and strain as we're gonna be using in the animal disease model. You guys have seen this slide before, so there are different routes of administration for our compounds. So I'm not going to be mentioning all of them, but what we have to have in mind is what is your drug for? What is the population you're going to be giving this drug? Here at the DDU, we work with neglected disease. So we really want a drug that you're going to be giving for the patients orally most of the times. So that is what you're going to be focusing, giving a drug orally, because then you don't need to go to the hospital and all the costs that add up when you have to be in the hospital to be taking a drug intravenously or by different ways. But to be measured how much of the drug we've been giving and reach the circulation by the oral route, we need to have our 100%. And our 100% will always be the IV, the intravenous dose. So here I just highlighted that we're going to be talking the next slides, okay?
Erika?
Yeah?
Yeah, just before you continue on, so
Yep.
it's a question about using the same-sex animals.
Mm-hmm.
Is it usually males or females? Because in Gemma's case, it was always males to avoid hormonal interferences, with obvious consequences on studying diseases always in male animals. And also, when in-house breeding, isn't this a problem where one of the sex will be discarded?
When it is breeding house, we're not going to be discarded this animal and it's going to be used for something, for control blood, for instance, and this kind of stuff. So we're going to try to use all the animals we have. The difference about sex depends from institution for institutions. So a lot of people would not use female because the hormones. But very recently, Charles River just published a paper saying that actually the female would be the best model to be using because they are more docile than the male. So they are less aggressive. And if you are working with a disease that there is no treatment, you really don't want a mouse to be aggressive with you. And the hormones you're going to measure down in the line as well. So here is the very beginning to see what the drug is doing and what your body is doing with the drug. But yeah, depends from institutions. In Brazil, I used to work with male all the time. Here, I work more with female, and depends. You're going to be testing both, actually. And actually, I just had a meeting today in the morning that some of our funders that give the money to us to do our research, they are asking to all our drugs to be tested in both sex in the beginning. And that can be a problem with the NC3Rs that I just mentioned, that we try to reduce the number. So if you're going to be testing both sex, you're going to need to have 6 animals instead of 3. So we don't know how it's going to progress. And we're going to need to have a lot more space in the animal unit to make the males ones in one side, the females one in the other. We normally don't put them in the same room because the pheromones, they can start to fight and you don't want animals fighting, even though they are in different cages and this kind of stuff. But just some new information came up today in my meeting in the morning. Yeah.
That was it. Thank you very much, Erika.
So entering the body. So how the drug get into the body orally? How does it get in there? So we have here our pill. I'm gonna be taking by mouth. You're gonna go to our stomach where it's gonna be dissolved. So here we need to have a drug that has a good solubility as we would have measured in the in vitro assay before go to the in vivo. And if your drug is not soluble enough, you're not gonna be going in vivo. Then you're gonna keep traveling, you're gonna go to your guts, and your drug is gonna have to be permeable. So you're gonna be able to travel to your guts, eventually reach your liver and not be destroyed by the liver because the liver is gonna try to destroy your drug as much as they can before reaching the systemic circulation. So I think it's very clear that the amount we start here, we're not gonna be the same amount we're gonna end up in here. So let's just put some number just to make something easier here. So if you are starting here with 100 mgs of a drug, you're going to be traveling, you're going to be losing 10 here, 10 here, 10 here. You're going to end up with 70%, because that is the percentage that you're going to reach your systemic circulation. And what I mean by systemic circulation is straight in the blood, is what is there in the blood that I'm going to be able to measure when I take a blood sample from my animal. And then you may ask, Erika, can you control any of these factors with drug? And I say, no, I can't, because I am a DMPK scientist, but the chemists, they can. They can make your drug more soluble. They can add something on it to make it more soluble, or they can make it more permeable or less permeable. They can try to avoid the liver to destroy your drug, to have a bigger amount reaching our systemic circulation. However, to measure how much of the drug you start giving orally and you end up in your systemic circulation, you need to know what is your 100%. And to know your 100%, you need to do the IV administration. So the intravenous administration is straight in the blood. So that's all you're gonna have. All in the blood is your 100%. Okay? Remember that I said about solubility for the in vitro test. But here, before we put a drug in the animal, we're gonna do another solubility test, because your drug cannot go into the animal in an organic solvent. Imagine if you are dissolving your drug in ethanol, for instance. You're gonna put 100% of ethanol in your mouse. You're gonna make your mouse drunk. It's not what you want. We want to try to find a soft solution here to administer in your animal, but it has to be, you have to be able to see that there is any particle in this compound. Because if you have a kind of a particle and you're going to inject it straight in the vein of the animals, you may block it, and then you don't have the blood traveling around your animal, and you're going to die. So you have to try to make a soft solution to inject into your animal. So here we try to use a little bit of an organic solvent that can be the MSO, methanol, ethanol, and you try to put a little bit of PEG or water if it is possible, and try to make it as soft as possible. So how it actually works. So just to have an idea here, we have our animals. So 3 animals for the PO leg, 3 animals for the IV leg. We're gonna make a hole here in the tail, and you're gonna take a few amounts of blood. So in this case, we took around 24 hours. So we divided these 24 hours, they contain microliters from 5 minutes after administration, 15 minutes, an hour, 2 hours, 8 hours, 24 hours. We mix with some water, so the bioanalysis team, they have a higher amount to be working with. And you're gonna just do a protein precipitation. So all the things that is inside of your blood that you don't need, you're gonna precipitate it, and you're gonna get your supernatant and injecting our high resolution mass spec that I mentioned. From the mass spec, we're gonna have all our numbers that you're gonna do all our dilutions and calculations that being made, and you're gonna end up with our graph. So here is our IV leg of the stud, and here is our PO leg of the stud. But something that I wanted to mention here is all the time that you see a graph like that, just remember that some animals have been used here to be able to generate this graph. So sometimes people would ask, oh, can you repeat this experiment? Because we have a point here that, we don't know what it is. No, you really try to make the most of what you get. You don't have to use another life for your experiment. So needs to be treated with attention. And, you don't want to be killing animals for the sake of it, because it's not nice. No one likes to kill them. That is what it is at the end of the day. Let's talk a little bit more about it. So here's our PO profile. And we have here our absorption phase. So it starts from 0. And then remember, it goes to your stomach, where you're going to be absorbed, just if it is at a solution. And you're going to reach a max. And this max is known by our maximum concentration, also known by Cmax. And the time it takes to reach our maximum concentration is our Tmax. Then it's gonna decline with the time, that's our distribution phase, and then eventually it's gonna be eliminated. And here, just to mention, there are different ways to eliminate a drug. So if you guys want to type in the chat, or if you want to unmute yourself, can you guys tell me which way you think the drugs are eliminated from the body? So drugs can be eliminated by urine, for instance, is a way. So your drug is traveling, go to the kidneys, you're gonna be eliminated by urine, can be eliminated by feces as well. They can be eliminated by breath, can be eliminated by sweaty. If you are breastfeeding, you know your drug can pass through to the baby if you are in that moment. So there are different ways to eliminate a drug. And I just put an image here again of our friend mouse here, just to think that when you have a graph like that, an animal, or actually 3 animals being used to generate this result. And this is an IV profile. So can you guys tell me if you can see any difference between the graph I showed before, the PO, in comparison with this one IV, apart from the color? Let's try to interact with you guys, because I feel that I'm talking alone. And no one wants to unmute and try to say something? I just put one slide back. So here, our PO profile, we have this phase here, that our IV profile, we don't. Anyone want to try to guess why?
We've got 2 in the chat. So we've got in the chat, we've got no absorption, and we've got the amount of blood concentration is higher.
So exactly, the difference here, we don't have the absorption phase. Remember, when we go IV, it's straight in the blood. So everything has been absorbed. So your Cmax here is your first concentration, is the maximum you're gonna get of this drug. So yes, well done for you guys that replied that that's the difference. So here we have our absorption. For the IV, no, we just have our distribution and our elimination. And of course, we have the AUC, that is known by the area under the curve, that's all the exposure of your drug. So it's everything that the moment that it enters the body until the moment it is excreted out. And again, just to remember that we have used 3 lives here to generate this graph. Just another concept that I would like to tell about you guys is, I have mentioned before in the questions about our MTD studies. That's our maximum tolerated dose of our animals. When you start to work in a project, we need to demonstrate that it works in the animal. It's not that we need to, but there is this expectations of the funders that, we're gonna prove that it works. So we need to have a good disease model traceability as well, to make sure that your animal predicts what you're gonna be seeing in the humans. Otherwise, it's kind of pointless. And if you see an efficacy in your animal model, you're going to be thinking, what is driving your efficacy? It can be driven by your maximum concentration, it can be driven by your exposure, but it also can be driven by your MEC. And I haven't mentioned this before, I'll be mentioned now. So MEC is our minimum effective concentration. So that's the concentration you're going to get from the biologist that you're going to give in vitro. So again, let's use the example of leishmaniasis. So my friend here, biologist, you're going to come and say, listen, this is the amount that we need to have to kill 99% of the parasite. So the profile of your drug would have to be above this MEC. And if you have a profile like this, remember, it's our oral profile. If you have one profile like that, it didn't reach the concentration that you need to kill your parasites. So probably your animals are going to still be sick, because you're not reaching what you need to kill 99% of your parasites. On the other hand, if you have a compound that is a lot higher you're going to be passing your maximum tolerated concentration. So above this, your animal's probably going to be showing some symptoms that they are not tolerating the drug you're giving. So you don't want that because it means that your drug is going to be toxic. So what you want is to be in between, so in the middle, above MEC and below MTC. That is when your drug probably you're gonna be working. So it's that window that we always want to be, above MEC and below MTC, okay? And if you have some problem here, if you have done this kind of experiment and you still think that your drug is not working, you're gonna, what is the problem? Because I am above my MEC, I am below my MTC, and why it is not working? So then it can go back to the biologist team and see if there is any problem with the biology of your parasite instead to be a DMPK kind of problem. And I will give some example in the next slides about it to you guys. But of course, you need to have an optimal study design. So our animal disease models are usually mice, but sometimes can be rats. And I mentioned before, rats, they are bigger. So if it's rats, you're gonna need to check with your chemist first if they would have the amount of drug enough to be tested in rats. So something always to keep in mind when you're gonna be testing a drug in the animal model. When you're going to do your PK, you're going to start with a low dose. You don't want to administer 100 mg of compound in your animal because you really have an idea what may be happening from your in vitro results, but you really don't know what you're going to be happening in vivo in your animal model. So you start with a little bit, 10 mg if it's PO, because you're not going to have 10 mg at the end. But if it's IV, it's even less, maybe 3 times less, 3 mg or 1 mg to see where you are. You need to think about the optimal study design. So how many times you're going to be giving that drug? Once a day? Twice a day? Remember that I said that the animal, the liver blood flow is a lot faster, 6 times faster in mice than it is in humans. So mostly likely in this preclinical area, you're going to be giving your drug more than once a day, which means coming to the work in different times of the day, sometimes during the night as well, just to try to have this regimen of dosage that you're going to keep your drug above your MEC that is killing 99% of your parasite. If your drug, for instance, is not working by PO, oral leg, you're going to try to do another leg. You can try to administer this intraperitoneal, subcutaneously, if it's something you can do in the project that is not restricted to BPO. Sometimes can be done, sometimes can't. And I mentioned before, we always try to take our blood sampling. So the main point here of this slide is we need to try to understand the relationship between PK and PD, and as fast as possible to try to make this drug as fast as possible in the clinic. But here we are trying in a mice model, and we don't want to optimize this drug a lot for the mice model because we actually want to give it for a human in our department here at the DDU. So can be tricky, but that is what we try to do.
Erica?
Yeah.
We've got just a couple of questions. So the first one is, does the MTC change between diseases? For example, for a cancer drug, you can accept some degree of toxicity. Who determines the MTC?
So yes, that's a good example because for cancer, your drug is going to be a little bit toxic regardless because the cancer treatment, you're going to try to be killing the cancer cells, but normally you're going to be killing your health cells as well. So yes, this is going to be a little bit more toxic if it is a cancer area. So what determines your MTC? So you have your maximum tolerated concentration and your maximum tolerated dose. So the concentration would be measured in vitro, the same as the MEC. However, the MTD, that is maximum tolerated dose, you're going to be measured by the animals. So what we normally do. After the PK, we do another experiment that you're going to take 5 days more or less, or depend how many days you're going to be giving this drug in your treatment. And you're going to be giving a mix starting with, and see how your animals behave with mix. And take spot blood as well and analyzing to see how much drug you have there. The next day, you give 30 mg and see where your animals are on it. Next day, 50 mg and see where you are until you reach 50 mg, 100 mg, and you can say, look, my animals, they seem fine. They are not showing any stress or any signs of toxicity. And we have our signs for the animal for toxicity. For instance, I can mention a couple here for you guys. So sometimes the animals, they walk in tiptoes. So things that they are not very comfortable with that drug. So that's a sign. So if you see something like that, we stop the experiment right away. We take the blood you need to take and you terminate the animal because you want to avoid any suffer. Or sometimes they have the ears a little bit down and things that they are not happy about it, or the eyes are a little bit down as well. So when you work with the animals, you can see the symptoms that they may present. And you have some charts all around the animal unit that if you see an animal in this way, or if you have some questions like, I don't know if my animal is good or not, you always have the animal unit people that you can just come and say, listen, come and have a look at my animal because I don't know if it's worth to stop the experiment, and terminate this animal, because if the animal is suffering, you don't want to make any suffer for the animal and just finish the experiment right there. Or if it is not suffering, something that you can give, I don't know, your animal is vomiting, for instance. So can you give another pill to try to avoid this vomiting and, improve the life of your animal, and this animal can still be accountable by the end of the experiment. So it's a line that you're going to be experiencing it when you work in this kind of environment.
Excellent, thank you.
I just have a sip of water. So, okay, I was saying before then about the doses. So how many times you're going to be dosing your animal? So we have some example here just to put a picture, in this kind of experiments. So as you can see here, just forget about this C90 and C50. It's same thing as the concentration that kills 50% of your parasites and the concentration that kills 90%. But what we really want is the concentration that kills 99% of your parasites. And if you see here in the graph, you can see this is your animals, right? It's the blood samplings of your animals. And here, after 4 hours, this concentration was going down the line, so was not killing my parasites anymore. And when we put this in the animal model of the disease, we couldn't see the treatment. So then my boss just thought, maybe because it is dropping after 4 hours, so we need to keep dosing these animals every 4 hours. So he asked the in vivo team to come to work and dose these animals every 4 hours. And guys, can you imagine? It's not fun. It's very unsociable hours of working because it goes through the night. But we did it, we dosed these animals, and we were able to maintain all the animals here above the EC99. However, we still not seeing the treatment in the in vivo model of the disease. So this compound went back to the biology, and then we said, listen, it's not a DMPK problem, must be some kind of biology problem. And that was very helpful in our process of drug discovery because this compound was a static compound, just static the animal. So it stopped the growth, but not kill the parasite. And what we were looking for a cidal compound. That's the compound that kills the parasite. So that was a good learning process for us. I will give you another example here because, sometimes you cannot come and dose the animals every 4 hours, or if you have another compound that, goes down EC99 every 2 hours, it's not possible to come and dose the animals every 2 hours. So we have tools to improve this kind of experiments that we can use. And I will just give an example here about the AT-hum. That is actually Lauren's favorite kind of animals. So don't worry about any of these results in here. But just to mention here, these are in vitro DMPK kind of results, and you use a kind of traffic light system. So when it is green, good, go ahead with the compound. When it is orange, yellowish, just treat the data with attention, more caution. And when it is red, is no-go. So we have all of these compounds in here that they were green, some orange here, and this were no-go. This was a particular clearance, so is what I mentioned before. The liver was just clearing this compound just too fast because the liver blood flow of an animal is a lot faster than humans. So what some people here in the university, they managed to create was a 80 mouse model. So they managed to replace the enzymes that the name we call them P450s. And they managed to replace these P450s from the mice by P450s from humans, which means that you're going to have a lot slower clearance when the CYP that is inside of the liver is from humans and not for mice. So yeah, when we did it with this 80 model, it was a lot. This is our cutoff for the clearance in the liver, and it was possible to progress with this compound if before wouldn't be possible. So, you know, remember what I said, at the end of the day, we want to make a medicine for humans, but we need to prove that it works in the animals. So here we are proving that it was not working in the animals, but we overcame this with this 80 model, okay? And this allows us to go to in vivo PK in 80 and welcome wild type as well. So here, just to show as well, it's the same compound in black. This black dot is the wild type animal. So this was a C57 background. And you can see here our lines, that is our EC99, EC90, EC50. If we were using the wild type model, we are not going to see any efficacy in the animal model. But if you are using the 80 model, we could see the efficacy. And then we were able to progress to the efficacy. And here is our animal model. Okay. I will just talk very briefly about our efficacy models here at the DDO. So we work mainly with 4 diseases. So it's Chagas disease, human African trypanosomiasis, animal African trypanosomiasis, and visceral leishmaniasis. And here I just put a photo of the Chagas disease animals that we work with, and I will explain this model further in these slides, okay? We start to work here, we used to have a problem with the animals because to be measuring the Chagas disease, to be seeing if the treatment was working, we needed to terminate the animals. Because Chagas disease, the parasites, they go inside of the organs, so inside of the heart, inside of the gut. And to be able to measure the treatment, we need to kill the animals. As you may imagine, it is not in agreement with the NC3Rs and all the legislation we have. So we needed to try to improve this model. And the way we improved this model was using luciferase expressing transgenic parasites. So very briefly, this is a firefly, and you know that they have this bioluminescent signal, and we managed to modify this genetically and put this in the animals. So remember last talk by Lauren, she was talking about the lock and the key. So it's pretty much that we need to use luciferase and we have the luciferin, and when they bind, you're gonna have a signal of our animals. And this, of course, we enter in agreement with the NC3Rs because we are able to not kill the animal to evaluate the disease, but we could follow as well the animal from the acute phase to the chronic and follow all the process of this animal. So it was a massive improvement in our lab for that. So how does it actually work? So we have our parasites that normally they are frozen, then we infect them in some RAG2 animals. These RAG2 animals, they are immunocompetent animals. So they just have an immune system a little bit more fragile than the bulb-C that you're going to use. And then the trypanosomes are going to be able to reproduce a lot faster in this kind of animals. We use just one animal for that. Then we terminated this animal. We purify the blood to take the trypanosomes and we infected our animals. After infecting our animals, we're going to do our treatment and we're going to evaluate by imaging, but also you're going to see how much of the drug is is in the animals, okay? So here, just to give you an idea how long this kind of experiments takes. So here is the acute. That is not what we really want, because the drug that is in the market kind of works for the acute phase. But as I mentioned in the beginning, no one knows that they are infected with ticrocide in the acute phase. So what we are looking for is for the chronic phase of the disease. So we have our animals here, and all of these blue arrows is when we do the images. So we do the peak of parasitemia, so we can see our animals very colorful here. We wait until around 100 days. That is when the animals became chronic. We do the drug treatment. We keep imaging the animals. We immunosuppress them to put the immune system a little bit down to see if any remaining parasites that is in any organs can just come up. We keep imaging them, and at the end, we're going to do our ex vivo imaging. So the ex vivo, I haven't mentioned it. Apart from now, ex vivo is pretty much when you have your in vivo system, and when your in vivo finishes, you're going to terminate your animals, but you're going to take your organs. So you're using everything of that animal. So just to put this in color and images for you, this is a kind of our experiment. So our experiment consisting around 36 animals. We always run the vehicle. The vehicle is what you use to dissolve your compounds in, just to see if it didn't treat. And we also run together the standard drug. So in this case, it's benzimidazole for the Chagas disease. This is the peak of parasitemia. Then we wait until they became chronic. I said this around day 100, but here, for instance, it was day 145. And we keep imaging the animals. And what we want to see is this: animals are clean. And even after the immunosuppression, as you can see here, we have some cleans, but some relapsed. But what you want to see is how the animals clean. Okay? If the animals are clean, we're going to do our ex vivo. So we terminate the animal and we take out the organs, and then we image these organs. And if we see any signal in any of the organs, for instance, in the heart, in the liver, in the spleen, in the lungs, in the guts, this animal is not considered clean. Okay? We had some problems before because we were imaging these animals and we couldn't see any signal. They were, any signal. And then we put the immunosuppression, and we started to see some signals. So these animals, they were hidden someplace. So we start to think where these animals are hiding. And as you can see here, an image, we could see some signal in this one. So where they were up to that point, up until we put the immune system a little bit down. And then we did some histology, and we could see some nest of the parasite. Okay, so we start to see these parasites in the blood as well. And as I mentioned, it's not expected to see these parasites in the blood. So why we see these parasites in the blood? We didn't know. So we start to have more questions than answers. So why we are able to see the signal during the chronic phase? You're not supposed to be there. It's supposed just to be inside of the organs. Did we catch up maybe these parasites whilst they were circulating? Where were they hiding? We didn't know. So with this in mind, we decided to do an ex vivo cascade. And for our animals be considered clean, our animals, they need to be clean on the dorsal, on the ventral. We take the blood from the heart, we take the brain, we take all the organs, and we image the carcass. And if at any point we see some signals in here, this animal is not going to be considered clean. Sorry for this. These images can be quite shocking for people that don't work with the animals, but just to say that we use everything of this animal. This animal has been giving his life for science, and we're going to try to use everything we can to get more information for future studies as well. So here, just put another example. So just to put all together, our vehicle, our standard drug, our compounds, and what we want is clean animals. The ones that are not clean, as you can see here, we're going to just terminate, not going to use. The ones are clean, go to the ex vivo, and that is the way we do for Chagas. Okay? I just mentioned very fast now about African trypanosomiasis. that can be either human or animal. We have the model as well. We don't have a bioluminescent model for this one, but in my point of view, there is no need because these parasites, they are in the blood. So it works a little bit like the ticrocide. So we have the parasites frozen. We don't need the rag tube because these animals, they can reproduce the trypanosome very fast. We infect them, we do the treatment, and we count the parasites in the blood. I have a video in here that I don't think I'll be able to play. Let me see. Oh, there you go. If you guys can see here, these kind of balls are the blood cells, and here are our parasites. So if you can see the parasites, it's because the drug is not working. If you cannot see the parasites, or if you can even see them dead, it's because the drug is working. So we don't really need a bioluminescent model for that. Okay? Oops. And now I'm gonna be playing again. So, and that is pretty much it, what I had to tell you guys. Of course, I will be expecting more questions when you finish this presentation, but just to finish here to say that there are a lot of people involved when we need to work with the animals. And it's something that is necessary to progress with our compounds. We need to put into the animals to have an idea or to know what to expect when it goes to humans, okay? If this is our summary, that is all that we've been through. So why the animals are needing for research, who are the scientists that work with the animals, and I think I made it sure that the animals, they have the rights, and we try to minimize the suffer as much as you can. And yeah, thank you very much.
Thank you very much for that, Erica. Whilst you take a drink, we have a question in the chat. So there's quite a bit of chat on this, which was asking about what happens if an animal cannot be infected. For example, HIV or COVID are quite specific to the human. What do you do in these particular cases?
Sorry, just having a sip of water. Yes, they are more difficult, but we have a project that we work in collaboration here that we can infect animals with COVID, for instance. They can be more tricky sometimes, but we need to try to find a model. And maybe if it is not infect, for instance, mice, it's going to be infecting dog or sheep. For instance, we have some experiments that we run in sheep here as well, that for schisto. So we just try to find the best model for your disease, pretty much.
Excellent, cool. And we've got another question as well. Can a person be doing DMPK science without getting hands-on with the animal testing parts?
Yes, they can. So the in vitro part of DMPK, you don't really have hands-on the animals. So the DMPK discipline is pretty much divided into three. So we have the in vitro DMPK, in vivo DMPK, and bioanalysis. So you can either be working in vitro or if bioanalysis that you're not going to be having your hands on animals, but all your results you're going to generate around it.
Excellent, thank you. Does anybody else have any more questions for Erica? I was going to say you can unmute quite happily, or you can put into the chat. So, Erica, you seeing the chat, or do you want me to keep reading them to you?
Yeah, I can read it then. I just need to catch up to see where you want.
Oh, just at the very bottom.
Yeah, that is humanization comes in like humanized mice. Yeah. Use of organoids getting popular in DMPK experiment or is this very experiment? We don't do, or well, I cannot say we don't do because we have done a little bit. One of the people that work with us, she had worked with organoids, but I cannot say much about that because I don't have much experience on it. You need a lot more stuff to be working with organoids, so it's not just the organs. You need to get some fluids from the brain and this kind of stuff. So I can't talk much about that. Like bionic organs. Yeah. We don't do much editing here, so I don't have much experience to be talking about this kind of thing. Sorry for that, guys. And any more questions?
Excellent. So, I mean, if you didn't get a chance to ask your question during the session, I mean, Erika and myself were both present on Slack, so you can ask as many questions as you like on the Slack channel, and I'll do my best to feed them to Erika. And yeah, I'll say that's today's session over. So that's session number 3. So yay, we've made it this far, which is really good. And we are going to meet for our last session, which is going to be a week today. So we look forward to seeing you. And as always, I will put the recording onto the YouTube channel and you'll be able to view it either by the end of today or tomorrow. So yes, so thank you everybody. So you are.
Thank you, guys. It was very nice to be talking to you today.
Thanks so much.